Great. We'll just give it one more minute. Okay, so it's past, so we will kick off. So it's a real pleasure today for me to introduce Sophie Sanford. So Sophie is a PhD student based here in Cambridge, and she's working in the UK DRI, so the Dementia Research Institute, in the lab of Dr. Will McEwen. So Sophie did her undergraduate in UCL in immunology and infection and has continued working within the field of immunology since then, and currently doing her PhD, where she's exploring the relationship between the antiviral innate immune system and the accumulation of tau protein in models of Alzheimer's. So Sophie is very involved also in scientific outreach and communications, and recently organized and chaired a virtual Making It Brain 2021 mini conference within the UK DRI. And this was giving secondary school students an insight into dementia research and the careers that are available. So Sophie's not shy of doing these types of events. So it's a real pleasure to introduce you today, and I'll hand straight over to you to start your talk. Thank you very much.
Thanks, Anna, and thank you very much for the invite. Yeah, I will just give a kind of overview of Alzheimer's disease and what I'm actually studying, and then my specific PhD project and some of the data that's come out of that. And yeah, I would really love if anyone wanted to ask questions either throughout or at the end. Really happy to answer questions, and also if you have suggestions for me, that would be really useful. So yeah, so an introduction to Alzheimer's disease. So we know that when you have a post-mortem brain, so after an Alzheimer's disease patient has died and you can take a look at the brain, we notice that there's a few things, and one of the things is atrophy. So that's where the brain shrinks due to the death of the neurons, due to the death of the brain cells. But some other things which are really characteristic of in the brain of an Alzheimer's disease patient are these amyloid beta plaques, and these are extracellular, so outside of the cells. You have these accumulations of amyloid beta protein, and they're called amyloid beta plaques outside of the cells. What you also see in Alzheimer's disease patients is these neurofibrillary tangles, which is these accumulations of tau protein inside the brain cells, inside the neurons. And so these are called like the hallmarks of Alzheimer's disease. So our lab looks at the neurofibrillary tangles, the tau protein accumulations. And one of the reasons we look at these accumulations is because they correlate very well with cognitive decline. So what is really interesting is that this is a graph showing a cognitive score done with a psychological test. And it's correlating that cognitive score of a patient with the amount of neurofibrillary tangles, or those accumulations of tau protein in the brain. And what you see is that as you have more tau or more tangles of tau, of accumulations of tau protein in the brain, you have a worse cognitive score. So we're really interested in trying to reduce the number of these tau tangles or accumulations of tau protein, trying to stop their increase, and therefore hopefully have an effect on the cognitive score or the cognitive status of a patient. So there's a really good reason for looking at these tau protein accumulations. So tau protein is a protein that's involved in the microtubules of the cell. And what that means is that it's a protein that's involved in stabilizing what we can think of as like the train tracks of the cell. So in a brain cell, you have to get nutrients and various machinery from one side of the cell to another. And to do that, you need some kind of train tracks so that they can be transported along. And so these are called the microtubules. So they're like little tubes that can that connect up one edge of the cell to the other. And tau protein is kind of known for stabilizing those microtubules. So when you look inside a cell, you can see tau protein along those microtubules, the tubes that connect either ends of the cell. So it's responsible for stabilizing that. But one thing is that it's really like spaghetti-like, so it's very disordered, and it's very easy to form these neurofibrillary tangles or accumulations of misfolded tau protein. So it comes off of the microtubules, comes off of the train tracks, if you like, and then it forms these accumulations of lots of tau protein. One thing that's really interesting, and that's been quite well understood now, is that these tangles, accumulations of tau protein, have this prion-like spread. And so what do we mean by that? So that means that when you have an accumulation of misfolded tau protein that has come off of the microtubules, it can spread its misfolded conformation and make normal tau, which is not misfolded, into more misfolded tau. So in this schematic that my supervisor made, you can see this tau protein accumulation of misfolded tau enters the cell, and what it does is it causes the conversion of normal soluble tau in the cell into more misfolded tau protein. And these are the neurofibrillary tangles that we saw in the brain, and then also those are the ones that correlate well with cognitive decline. So there's a nice analogy for this, there's a nice model, and it's kind of based on this novel called Cat's Cradle, where in this story there's a crystal called Ice 9, and it's a solid, and when the Ice 9 solid touches any water, it makes that water into ice. So you can therefore carry some of this solid with you, and then that will turn the lake into ice or something, so you could walk across it. So it's kind of like that, where we have our misfolded tau protein that touches and accumulates those normal tau proteins, and then they become misfolded too. So that's how we understand the tau aggregates, the tau assemblies, to spread throughout the brain and therefore accumulate and possibly therefore contribute to cognitive decline, or are associated with cognitive decline in Alzheimer's disease. So I've explained this process where you have the misfolded tau protein and it has to enter the cell, and then it causes the soluble tau to misfold. And what my supervisor is really interested in is the fact that this process is very much like the way a virus has to get into a cell. So much like the tau aggregate, the tau misfolded tau assembly, a virus also has to enter a cell, and it also has to replicate inside the cell. And that's the way we have a viral infection. Obviously, it's very relevant at this time. But the thing is, our body has natural innate immune defenses against this process. So it's not an easy process for a virus to get into a cell, because our bodies are made to have an immune system against this. And yes, we have antibodies and things like this too, which are produced later on in the infection. But we also have things that are inside our cells that are there as soon as for the time as soon as a virus enters a cell, so we're ready for it. And what happens is, when a virus enters a cell, the cell produces this immune signaling molecule called interferon, and I will now call it interferon, and I might call it a warning molecule from now on. And it's basically just a warning molecule that the cell releases. It's an immune molecule saying to neighboring cells or cells next to the infected one that there's a virus in here, you need to start upping your immune defenses because we need to get rid of this virus. And this happens all the time because we're probably coming across a lot of viruses and other pathogens. And then this actually helps us control the infection. So what happens is that interferon attracts immune cells to the area. So this warning molecule brings in immune cells to the area where you have the virally infected cell, and it causes flu-like symptoms. So when you feel flu-y, when you have a viral infection, one of the main reasons is because you're producing so much interferon. It's because it's attracting the immune cells, and it's doing that by increasing your blood rate into the area. So you might feel fevery, and that's really due to you having a lot of interferon warning molecule. And this stops the spread of the virus because you have a lot of interferon stimulated genes or factors inside your cells, which cause the virus to be destroyed. And there's about 500 of these interferon stimulated components which are produced in response to the virus. So this brings me to my question. I've talked about how interferons can restrict virus spread and viral replication, but can they also restrict tau aggregation? And that was the question that really overarches my PhD, because we know that these natural defenses are in there anyway for viruses. So do they have any influence in tau aggregation and tau protein as it comes into the cell? I'm just mentioning this now because I think it's quite interesting, which is the importance of interferons in COVID infection. And so I mentioned a virus, when a virus enters the cell, it causes interferon production. And this is the same for the current SARS coronavirus. And what's really interesting is that people who have very, very severe COVID infection have been analyzed in hospital studies and things like this. And what they've found is that some of these people have anti-interferon autoantibodies. So normally we shouldn't have any antibodies that are against our own proteins. So we should only have antibodies which are against foreign things like viruses and bacteria. And this is done through your immune system as you develop. But what has been found is that people who have really severe COVID infection have antibodies that are removing the natural interferon. And therefore, these people just don't have as much interferon as people who have an asymptomatic infection or something like this. And therefore, the interferons can't restrict viral infection anymore, and these people get very severe COVID infection. So this is one part of very, very current research which people are quite interested in. So to break that big question down, does interferon affect tau aggregation if it can restrict a virus? I'm investigating the effects of interferon, this warning molecule, on tau aggregation, both in a dish, in cell, and on tau pathology in the long term. And I'm going to go through these two aspects that I've been investigating. So if we want to look at tau aggregation, we need to have a model of tau aggregation. And this model was developed by my supervisor. And basically, all we have is cells, immortal cell lines, and that just means we can use loads of them all the time because they never run out, because they divide all the time, and they're immortal. So we have these cells, and then what we do is we make them produce a green tau protein. So they look like this under the microscope, where we can see that they are expressing a green tau protein. And so they're overproducing this tau protein. And at this point, this tau protein is doing its normal job of stabilizing the microtubules or the train tracks, and it's not misfolded. But what we can do with this model is we can add tau assemblies or misfolded tau protein that we make ourselves in the lab. And this causes the tau to aggregate. So we can see these this green tau turn into these very bright tight puncta that now we can quantify, which is, as many of you might know, like a very useful thing when you can have a quantifiable number for something. So this is the difference that we're working with, and we count the number of spots using an imaging plugin, and then we can determine how much tau aggregation there is and whether we can reduce that, perhaps by adding interferon. So this was the first thing that I was doing. So as I mentioned, we put these tau assemblies on the cells, and that causes the green tau to aggregate that we can, in a way we can count. And what I would do is treat the cells with interferon or other aspects of viruses and bacteria, and see whether that reduces the tau aggregation induced by a tau aggregate. So can interferon reduce that process of tau aggregation induced by a misfolded tau protein? And what was really interesting is that I used two different types of cell. So the one on the left is derived from a brain cell. So it's derived from an astrocytoma, for anyone that's interested. It's basically the closest we can kind of get to a brain cell for a human. And these are all human-based cell lines. And what I did is I put more and more increasing doses of interferon onto these cells, and then I measured the number of aggregates, the number of those green tau puncta that we count. And then what I saw is that interferon actually increases tau aggregation. So it doesn't reduce tau aggregation in a dish, it actually seemed to increase. And so that was one cell type, but I was doing them in parallel. I was doing it in a different cell type as well. And this is the normal one we use in the lab. And what I found is in another model, in another cell type, also human, when I treated with interferon, despite having good upregulation of all of the interferon stimulated components, and I checked this as well, there was no effect on tau aggregation in a dish. And we can kind of discuss why this might be, but essentially a lot of the immune signaling components of these immortal cell lines are a bit funny because they're cancer cell lines, and therefore they don't have a similar immune system as our working cells do. So we thought perhaps this isn't the right place to be investigating this in, because it doesn't have all of the immune cells that we know are so important for viral infection as well. So we decided to move into a different model. And what has been recently developed in our lab is a model of this tau aggregation, but instead in brain slice cultures. So what this involves is culturing a slice of mouse brain on a membrane, and that allows us to culture them for about 5 weeks. And this slice of brain, we take the hippocampus, so this is one region of the brain that we take it from. We put that on the membrane, and then again, these mice are transgenic, so that just means that we have given them a human tau gene, and so they're producing a lot of tau, these mouse brains. And that means that we can actually add misfolded tau proteins and watch tau aggregation occurring in a brain slice culture model. And there's loads of advantages to this. One being that in this model, we have lots of different brain cell types, so we can kind of see the way that they're interacting with each other as well when we're adding things like these immune signaling molecules. And so what I do is like take this slice culture, and at the end of the process, I'll image it on the microscope. And when I add this recombinant, so this is just tau misfolded tau assemblies that we make in the lab that I used for the other experiments too. This actually, you can see that this induces a tau aggregation and we can monitor this by staining for a phosphorylated or like, which is representative of misfolded tau. So basically, we've just got another way of recording tau aggregation, and it takes a lot longer and it's a lot more complex, but it's maybe a bit more representative of what's actually going on in the brain. And this was developed by Lauren and Amir in my lab, who I'll mention at the end. So what's really lucky is that we have access to 2 different types of brains, if you like. So some of the brains have the interferon receptor, so therefore can respond to the warning molecule. And then the other brains don't have the interferon receptor. So these are a genetic knockout, and that just means that these mice don't have any of this interferon receptor. So they can't respond to interferon. So even if it was there, they shouldn't be able to respond. And what I found is that when I add interferon to these brain slice cultures, and then I added a recombinant and misfolded tau assembly, firstly, if you compare this control, so that's no tau aggregate added to the seed, which is what we give the name for the tau assembly, you see that that tau aggregation, and that's the staining that I'm quantifying in this image, that's increased. But then when I add interferon, that increases it even more. So that actually kind of sort of fits with what I saw in this cell type, the first cell type, where interferon is not restricting tau aggregation, but is actually increasing tau aggregation. And so now you might ask, well, if you use brain slices that don't have the interferon receptor and therefore can't respond to it, we would hypothesize that this effect wouldn't be there. And that's what happened. And in this circumstance, when I added interferon to brain slices that didn't have the receptor and therefore couldn't respond, there was no more increase in tau aggregation. So that's kind of pointing towards the idea that tau aggregation is being affected by interferons, and actually the interferons are increasing tau aggregation. So what about in the long term? So this is great. It's a 5-week model, but we know that Alzheimer's disease occurs over a much longer time period, and a lot of these neurodegenerative diseases. So can we get one step closer to what's actually happening in a human brain? And then, so what I'm then doing is going in vivo. So what we have is a mouse model of Alzheimer's tau pathology. So these mice develop tau, these tau neurofibrillary tangles that you saw in the image right at the beginning, and we can see that in the brain, and you'll see some images as well. So they're transgenic mice, so they express human tau protein and they develop this tau pathology. So we can monitor them over a series of months, and they develop this pathology. Again, I'm using these 2 different types of mice. So some of them have the interferon response working, and the other ones don't have the interferon response working. So they don't have the interferon receptor. And so now we're asking a slightly different question, but it's all along the same lines. And that's, if these mice can't respond to interferon, does this affect tau aggregation? So if they don't have this working interferon response, then is that increase in tau aggregation that we saw earlier mediated by interferon, is that still there, for example, or have we got rid of that by if they can't respond to interferon? And so these mice were aged until 6 months of age, and then I've been analyzing the brain samples from these mice and monitoring the tau aggregation. And that's the interferon receptor there I've put on the surface of the cell. So I just did a quick check to just make sure that these mice really didn't have the interferon receptor, and therefore, and what I found is that when I did a quick test for whether these mice had less of this very pro-inflammatory cytokine, it's just another word for a signaling molecule, I found that the ones that don't have the interferon receptor have much less inflamed brains. They have a lot less pro-inflammatory signaling molecule. And so that seems to be actually happening in these mice. So now is the question, is the tau aggregation actually affected in these mice if they have less inflamed brains? And so what I've done here is that I've stained the sections of brain from these mice after they were aged 6 months. And as you can see, this is a stain for the aggregated tau here. It looks a little bit different to the human staining, but it's pretty even. And this is the cerebral cortex at the front, the cerebellum at the back, and the brainstem that connects to the spinal cord. And as you can see from this data, there is a difference. And that was very, very exciting moment in the lab, because it was an idea and it actually did seem to show up in these mice. So I was very lucky that this phenotype came out. And what I saw is that indeed, in the ones that don't have the interferon receptor and therefore have less inflamed brains, they seem to have less tau pathology, and they seem to have less tau aggregation. And then what I last did, and to just connect these two models up, is that's just one way of looking at tau pathology, is by staining for the tau aggregates in the brain. And that's a really good way of doing it. But there are other ways of also just checking that they actually do have less tau pathology or less tau aggregates in the brain. One thing we can do is that we can take some of the brain homogenate from these mice. So all I do is I mash up, I mean, it's quite gruesome, but I take some of the sample of that brain, and then I can put it back on our model of tau aggregation in a dish. So if you remember in that model, those cells express the green tau, they produce green tau. And when we put these recombinant tau assemblies on them, we see the tau aggregate into these puncta. And what I can instead do is, instead of putting our own tau fibrils, tau assemblies that we make in the lab, instead of putting those on, I can put some brain sample from those mice where we saw the reduction in tau pathology. And I can just actually check, do those mice that don't have the interferon receptor, and that seemed to show up in the staining, that they have less tau pathology. Do they actually have less tau pathology and therefore misfolded tau species? And I can check this by putting it on the green tau expressing cells. So do they have less or more tau aggregation? And the really nice thing is, and I apologize for the data points. The nice thing was that it still is conserved and it correlates. So those mice that had very high tau pathology by the staining, that they also had really high numbers of aggregates that I could quantify in those. So they had really high numbers of these. Again, those mice that had low staining and low tau pathology also had low numbers of these assemblies, of these puncta. So that was a really nice way of just checking that they actually did have less misfolded tau assemblies. And what I'm now going to summarize is that in cells, interferon had variable effects on tau aggregation. And we started out, thinking it might restrict tau aggregation because it restricts viral infection, but actually something different is happening here. And in brain slices, interferon appeared to increase tau aggregation. And this really seems to fit with our model in aged animals, in these mice that develop tau pathology, where when these mice can't respond to interferon because they don't have the receptor, they have less tau pathology. And so interferon really seems to be pointing for a role for interferon in increasing tau pathology and contributing to the neurofibrillary tangles in the brain. So I'm just going to tell you some future plans, because obviously this brings up a lot of questions. So why does interferon increase tau aggregation? I'm trying to work it out. Your guess is as good as mine. Do the immune cells of the brain play a part in interferon production? And do these immune cells also, are they changed by interferon? It's probably yes. And I'm just trying to work out the best way to analyze these immune cells and see how they're affected by interferon and how they contribute to tau aggregation. Another question is, why is interferon important in the first place? And is it that actually, if you have more tau aggregation, does that cause interferon to be produced? Like, what is the source of the interferon? Because we know it's induced by viral infection, but could it be from other sources too? Could it be from tau aggregation? Because we know that Alzheimer's disease patients do also have kind of inflamed brains as well. Again, do the mice without the interferon receptor have less inflammation? And is this the reason why we have less tau pathology, less tau aggregation? So these are the questions I'm trying to answer now, as I have just entered my third year. And with that, I would like to say thank you very much to my lab. I have a really, really great supervisor. I can really recommend him. And have a really nice team. And Lauren Miller and Armin Mikadam are the two people that developed the brain slice culture model of tau aggregation. Will has that back in the day made this green tau expressing model of tau aggregation. And we're all kind of working on lots of different parts of the innate immune system and tau pathology. And this is just one part of it. So thank you very much. And yeah, happy to take any questions. And thank you for my funding, Alzheimer's Society.
Amazing. Thank you so much, Sophie. That was a really fascinating talk and so clearly described as well. So thank you very much for that. So we'll kick off with the discussion section. So if anybody has questions, feel free either to post them in the chat, or you can just unmute yourself and ask the question. Perfect. We have one in the chat already. So this is from Professor Tetti, and she's asked, which cells secrete misfolded tau, and is it possible to block extracellular tau pharmacologically, for instance, by monoclonal antibodies?
Yeah, so this is actually something that our lab is working on. So that specifically that antibody immunotherapy. And I think so a lot of these monoclonal antibodies that can bind to the tau in the extracellular space before it comes into the cell, there are a lot of those trials going on right now. I think I know some of them haven't actually succeeded in reducing the misfolded tau assemblies. But yeah, this is something we are working on, and it is possible to block it. It's just whether it actually works in a human trial. I think that's actually the difficulty, it seems. And our lab's working on the mechanism by which it can actually reduce the tau aggregation. And which cells secrete misfolded tau? So I guess actually the only cells that express tau protein are neurons and maybe glia. I might have to double check, but yeah, definitely it's only neuronal. So it would probably be pushed out by neurons and also the surrounding glial, microglial cells, which are like the immune cells around that area.
Great, thank you. So while we're waiting for any other questions to come in, I'm going to be really unfair and ask you if you have a hypothesis as to why the interferon is increasing tau. So I know you said that's the last step of your PhD, but just to have an idea if you have any ideas as to why that might be.
Yeah, so there's like, we're just kind of trying to take clues from the literature at the moment, and that's included looking at things like specific type of glial cells which are affected by interferons. So for example, we know that there are like interferon responsive microglia, so in the brain that have particularly like heightened interferon response. And is it possible that because you have this basal level of interferon in the brain, these microglia are around, and therefore maybe they're contributing to tau pathology by I don't know, spitting out by engulfing neurons that might have tau pathology and then spitting out more tau, misfolded tau. And then maybe in our mice, in the knockout mice that don't have interferon receptor, maybe it's possible that we have less of those, for example. So it's now a matter of like trying to look at the basic things first. So is there actually just an increased amount of these glial cells of information, and is that just what's contributing, or is it there's no change and it's actually about the status of the immune cells that's affected by interferons? It could be to do with a specific interferon stimulated gene, for example, like it could be an actual component that's induced by interferon that's actually affecting tau pathology. But yeah, these are the kind of questions we're asking.
Great. Well, maybe this time next year we can have you back and you'll have the answer for us.
Try and work it out.
Perfect. So we've got another question from Oriana. She said, can you get the misfolded tau aggregates outside of the brain? And if so, does this cause disease?
Ooh, do you get outside? So I think you can see them in the cerebrospinal fluid, and these are like the targets of a lot of current like biomarker early detection tests for Alzheimer's disease. So you can get them in the cerebrospinal fluid, but the thing is that they're very, very low concentration. So I think yes, you can. And I don't know about blood. This is a like common theme of like, you know, if it gets into your blood, can you then spread tau misfolding through blood and things like this? If it is, I think it's very, very low. So I don't think at the moment, we don't think it's like a prion disease where if it was in your blood, it would cause prion disease. But I think, yeah, it would be low. And it seems that way at the moment. It doesn't seem to cause this.
Great, thank you. And another question here from Hannah. She said, are you aware of any other diseases where the severity increases with levels of interferons? And if so, are there any correlations between these diseases and Alzheimer's?
Yeah, that's a really, really good question. So a lot of people have looked at, for example, HSV, herpes simplex virus infection, and Alzheimer's disease, and people have, I don't know, it's a bit controversial, but like have correlated these infections with Alzheimer's disease. And we would know that that virus would induce interferon, and that might be at a basal kind of chronic level. And people have also looked at it in aging. So I'm literally writing a literature review at the moment, but we know that interferons generally in the brain increase with age. So the brain expresses more interferons with age. And people have looked at interferons in other models of neurodegenerative diseases as well. So prion disease, and basically it really seems to differ, the effects of interferon, depending on the model that you're using. But what's important is that in humans, we know that interferon, like transcript of the gene, that expression is increasing with age. And yeah, you get more of these interferon responsive glial cells with age as well, and things like this. So yes, and people are looking at like viral infection and if that leads to hospitalization, and then that correlating with Alzheimer's disease as well. So yeah, one really recent study was about multiple sclerosis even, and they've like really like hammered down that Epstein-Barr viral infection could be a causal thing for multiple sclerosis. So yeah, like a lot of it has been correlation so far, but then perhaps we're moving more towards causal inferences now. And it'll be really interesting with COVID as well to see. I was like really curious, maybe like, do those people who have anti-interferon antibodies, could we monitor them later on for Alzheimer's disease or something?
Yeah. So this was a question I was going to ask you, and at the beginning of your talk, the question was going to be, is there a correlation between these people with the anti-interferon antibodies and having COVID? And then as your talk went on, the question sort of changes to the opposite, right? So if you have these, if you sort of have this more severe COVID, maybe that's going to make you a little bit less susceptible or protected against Alzheimer's.
Yeah.
But I suppose that data is not going to come out for quite a while. I know with Parkinson's disease, they're looking at correlations already. Do you know if there's anything on the Alzheimer's story yet?
For like interferon antibodies
and Alzheimer's?
Yeah, no. And I actually asked someone who was looking at autoantibodies, and I asked if they'd done it in an Alzheimer's cohort, and they were like, we're waiting for a better cohort. So no.
Right.
Yes.
Okay. That'll be.
But yeah, I mean, it's quite big strides to take as well. Like this is, I would also preface, like my data is limited to these very like tau overexpression models of tau pathology, and I'm not going to say this is how it is in Alzheimer's, but it's in one model and it's one part of the puzzle maybe. So yeah.
Okay, and another question in here from Mauro. It says, very interesting and clean data with an excellent presentation. Congratulations. Keep going.
Thank you. It looks clean on there, but it wasn't clean.
Well, you're presenting it beautifully, so it's done the right thing. Okay, I do have another question while we wait for any more to come in. So one of the big questions, I guess, with any of the inflammatory neurodegenerative diseases is this question of what comes first. Is it inflammation that's causative to the disease, or is the inflammation arising as a part of the disease that's then perpetuating it? And I think your story is really interesting in this. And I'm wondering, with your mouse models, whether you've had any possibility to look at the interferon receptor knockout mouse model, which isn't an Alzheimer's transgenic, which you could then introduce the tangles later on and understand the effect of introducing the tangles sort of without having the parallel transgenic Alzheimer's and the interferon receptor, if that makes sense. So if you don't have the interferon receptor, what would happen later in life, to understand that cause effect type question?
Yeah, that's really interesting. I think the issue we come across is with that is the when if we were doing it in a non-Alzheimer's disease background, we can't actually look at tau pathology because it's too low. Okay. So you actually can't induce And actually people are kind of working on this, like whether you could try and induce this tau pathology in normal mice that don't overexpress the tau. So that would be what we'd come across. But that's one thing I really want to look at is yeah, I will still have this tau producing overexpressing background, but can I look at tau tangles, and if I add them to mice that don't have the interferon receptor, do they have a different response to tau, for example? Are they less inflamed in response to tau? And people have actually done this in those amyloid beta plaques that I mentioned at the beginning. So they've treated normal microglia or normal glial cells, and then interferon receptor knockout glial cells. And they actually have the ones that don't have the interferon receptor have a really dampened response to amyloid beta. So that seems to say one thing about interferon is actually important in the pro-inflammatory response to A beta. And this is something I want to do within our models as well.
Yeah.
So,
and presumably the role of, because interferon is a very pro-inflammatory cytokine, presumably that's also affecting the responsiveness of microglia to any sort of infection. So is there a concern if we were to look at this therapeutically and say, let's reduce interferon gamma receptor expression, is that likely to cause other issues?
Yeah, it definitely would. Yeah, I think it probably isn't the best idea to do it like really long term. So we are kind of looking at pharmaceutical inhibitors of that interferon pathway. So one of the drugs I'm looking at the moment is called baricitinib, and that's a JAK1 inhibitor. And that just means like the response downstream of the interferon plus interferon receptor is dampened. And that's already used in rheumatoid arthritis cases. So what would be interesting is if you're dampening this interferon response already for things like rheumatoid arthritis, are you also affecting risk of Alzheimer's disease? And people are looking at this too. So yeah, maybe it's not, you don't want to get rid of interferons, but it's just interesting to know that this is one thing that could be contributing. And
yeah, great. Fab. So do we have any other questions at all? Feel free also just to unmute yourself if you want to ask it yourself, or pop it in the chat. Just trying to translate what that means. I think that's a question of credits and not a question for you.
Cool.
Okay. Oh, one more here from Oriana. What is a prion disease?
Oh yeah, sorry. So prion disease is a disease which is caused by a misfolded protein. So instead of having a virus or something, it's caused by the misfolded protein. And this results in the typical prion disease results in brain neurodegeneration, brain degeneration. That's all caused by a misfolded protein, and that can be transmitted. So there's like a cow prion disease and things like this, and the misfolded protein can then enter the brain and transmit that misfolded conformation, and that causes the neurodegeneration. So that's like the kind of model we use for tau pathology. Yeah, it's different to genetic disease because it's caused by a protein and not and it can be genetic. So that's the complicated thing, because it can be genetic because you can have a mutation in the prion protein that makes it misfold, but you can also just get some misfolded protein without having any genetic issues, and then get prion disease and get prion misfolding. And there's some really, really like sad cases of prion disease developing in people that has been transmitted to even through research facilities and stuff like this. So we have to be careful also when we're looking at tau and stuff like this.
Okay, any other questions at all? Or comments or suggestions? As Sophie mentioned, she's got 1 year left. So for any PhD student, we're always very open to suggestions on the research.
Please.
And if you do, Sophie, if you're happy for me to share your email address, if anyone wanted to contact you afterwards, would that be okay?
Yeah.
Brilliant. Okay, in that case, if we don't have any other questions, we can wrap up here. So thank you so much again for your presentation today and for joining us. And we hopefully will have you again soon with us.
Thank you. Thank you. I look forward to the rest of them as well.
See you all next Thursday.